Thank you so much, Tony, and thank you everyone in the audience for getting up so early to learn more about antibody-based molecular imaging probes. So, first of all, I did want to just briefly introduce you to some concepts that actually will probably follow through the session, which is the choice of the class of imaging agents that you're going to use, and in particular, why we are working primarily using antibodies. And that's because, as most of you know, we are able to, first originally through the mammalian immune system, we're able to obtain antibodies quite readily with high specificity and high affinity to pretty much any protein targets that you're interested in. And those of you who are laboratory-based know that these are workhorses. We use them every day for assays to detect very specific proteins, ELISAs, Western blots, flow cytometry, IHC, etc. And in the clinic, after years of research and development, we also know that antibodies that recognize targets such as HER2 that's overexpressed in 20% of breast cancer can form the basis of very effective therapeutics, such as the drug trastuzumab. So they clearly have proved themselves as able to hit targets and also to hit targets in patients, importantly. And then just as a reminder, if we look at a report from last year, at the top 10 cancer drugs from the previous year in 2011, 4 out of those top 10 are antibody-based products. And many, many more are in development. And I think that's also a key point to keep in mind. This clearly is a proven path to targeted therapeutics. But as we develop more and more of these targeted therapeutics, there also is a critical need to have the appropriate molecular diagnostics so that we can understand which patients are appropriate for use with which targeted drugs and whether or not those drugs are actually working. So in parallel with targeted therapeutics, we have to have molecular diagnostics in order to be able to use them effectively. So what about imaging with antibodies? What if we could take immunohistochemistry? We see this beautiful staining here of a prostate cancer with an anti-PSCA antibody. What if we could actually take that into patients? Well, it has been done actually to do what I like to call in vivo immunohistochemistry or in vivo IHC. Shown on the right is a image from a publication by Neil Bander and his colleagues where they've taken this an anti-PSCA antibody, intact antibody, radiolabeled it actually with lutetium-177 and imaged metastatic disease in prostate cancer patients. Okay, so first of all, that's the concept, and I did want to touch on the question of what do we look for when we're trying to pick a target that's suitable for antibody-based imaging. And of course, the targets that are going to be of interest to us are things that are going to be overexpressed, elevated in the tissue of interest, the disease state of interest, or equally important these days, something that changes in response to a therapy, a targeted therapy in particular. So we need something that's going to have elevated expression. We also are looking for targets that have low expression in normal tissues so that we have little background. We also want, hopefully, targets that are not shed or shed at only very low levels into the circulation, because otherwise they will provide a circulating sink to capture your imaging reagent and make it a little more challenging to detect the target that you're interested in. If we're using antibodies, by and large, we're restricted to cell surface targets. And I'll say by and large because more and more, the rule may be, we have to look at cell surface targets, but then I think as you get more and more into the biology, people are finding exceptions, for example, in tumors where things that should be intracellular targets do end up on the cell surface. So that's something to bear in mind, to keep an open mind, but basically, to a first approximation, we're looking at cell surface targets. One thing we do have to pay attention to, and I'll talk a little bit more about this later, is what happens when your antibody agent engages its target. Does it stay on the surface or does it internalize? And this is important because it will drive your decision as to what kind of radiolabel to use and what kind of chemistry to use. And finally, circling back kind of to the first point, we do need to identify targets that are, it's a lot of effort to develop these for preclinical imaging and even more to develop these for clinical imaging. We have to focus on targets that are going to give us important information about biology and therapy. And if you look at a timeline of development of antibody-based products that have been approved by the FDA as shown here, you can see that we have I keep updating this slide, and I think I'm going to run out of space soon. You can see that throughout the years, the accelerating pace of approvals of antibody-based drugs and products. And the other thing I'll point out is if you look at the color coding, the black ones are just the unlabeled antibodies that are therapeutically active. The red ones are the approved imaging agents. The purple ones are the radioimmunotherapy agents, of which there are two, Bexar and Zevalin for radioimmunotherapy of B-cell lymphomas. And then in blue are the antibody-drug conjugates, and that's actually a field that is of great interest lately. It's taken a while to work out the technology, but tremendous progress has been made, and now we're seeing approvals of antibody-drug conjugates at an increasing rate. Excuse me. You'll notice that among the first imaging antibody-based agents that were approved, 4 of these were imaging agents, but they really have not done well. Only one of them is still on the market, and it really is has not seen widespread clinical adoption. And there's a number of reasons for this. First of all, please note that these were approved back in the 1990s, so we're talking 15 to 20 years ago. And these decisions were made, 5-10 years prior to that as to what targets, what antibodies, what radioisotope, what approach to take. So these really were first-generation products, and you can see that, in some cases, the target selection was not ideal. For example, prostacin, the antibody detects a cytoplasmic epitope of its target, so it really doesn't work so well on viable tissue, but rather on necrotic tissue. And then these first-generation products tended to be based on murine antibodies. So they were immunogenic in patients. If you had a patient with an intact immune system, you basically could only use it once because the patient would develop antibodies against your imaging agent. So that really limits utility. Another challenge with intact antibodies, they're great as therapeutics, and one of the reasons they are is because they have a long circulating half-life. However, if you're trying to image, this presents problems because you have a pool of radioactivity in the blood and the body that it persists for several days. And in fact, most of these antibody agents that are based on intact antibodies require you to wait 5 to 7 days after administration before you can then bring the patient back and do your imaging study. So, nonetheless, despite those challenges, that was then, this is now. And now there is renewed interest in using antibodies for imaging purposes due to some parallel advances in different related fields. One is antibody-based engineering. You'll hear more about that in my talk, because that's an area that we focus on. But first of all, to reduce that problem of immunogenicity by moving to chimeric, humanized, and fully human antibodies, and also the shift to using antibody-based fragments where you can tailor the pharmacokinetic properties depending on what your final application is. In parallel, there have been huge advances in clinical imaging. As you know, positron-emission tomography has now grown in widespread utilization, mostly using fluorodeoxyglucose for imaging in oncology. But what it means is that PET scanners, PET CTs are widespread, certainly in the U.S. and to a large extent in much of the world. So the infrastructure is there to image with PET with all the advantages it provides in terms of increased sensitivity, resolution, and importantly, quantitation, the potential to quantitate your imaging. And finally, I think one thing, and this is what, in the end drives what we're doing, there continue to be, in fact, there are growing unmet needs in medical imaging and in drug development for targeted imaging agents. And this is, again, the culmination of another long story, an antibody called CG250, which recognizes carbonic anhydrase 9, labeled with iodine-124 for PET imaging. This is actually, I think it was a multi-center trial, but Diffke is the lead author. This agent has completed phase 3 clinical studies as an ImmunoPET agent in renal carcinoma. It's highly diagnostic for clear cell carcinoma. And so we're looking forward to seeing this progress further. It'll be the first ImmunoPET agent that's moving forward into hopefully registration. So what are the challenges in ImmunoPET? So I just told you that, PET imaging is widespread. People are using fluorodeoxyglucose. The most widely used radionuclide for PET imaging is fluorine-18. It has a 109-minute half-life. So this is what I see as the central challenge to antibody-based PET imaging. And the question is, what happens when an irresistible force, the decay curve of fluorine-18 with a 2-hour half-life, meets an immovable object, which is, of course, an antibody that just sticks around for 2 to 3 weeks. So we've had to address this on both sides, we as a field. On the one side, people have moved more and more to using alternative positron-emitting radionuclides that have longer half-lives. I'm not going to go through this table in great detail, but to just point out to you that fluorine-18 is here with a 1.8-hour half-life. Gallium-68 is great because it's generator-produced, but it has about a 1-hour half-life. We have 3 here with intermediate half-lives of about between half a day and a day. And then there are 2 here at the bottom, zirconium-89 and iodine-124, that have half-lives of 3 to 4 days. So those extended physical half-lives open up the window for imaging using agents that take longer to get where they're going. But there are a lot of other additional considerations that you have to take in mind when you are selecting a radionuclide, including the positron yield. You can see it varies from around 20% to close to 100%. The energy of the positron that's emitted, and also these radionuclides that have modest positron yields also emit other stuff. So you have to take a look at the entire decay scheme and see what it does in terms of your imaging quality and also your overall dosimetry. So those are all things to keep in mind. Nonetheless, these are all radionuclides that people in the field have been pursuing with great interest for preclinical and clinical development. One other point we have to keep in mind, too, though, is how you attach that radionuclide to your antibody of interest. There are stable linker chemistries, there are labile linker chemistries, and which approach you select is going to be a function of whether or not the target you're engaging internalizes or not. And I'll just sort of briefly go through some of the considerations here. To simplify things, basically one can consider sort of the straightforward standard iodination procedure where you just oxidize and the iodines get added onto tyrosine residues. That's actually a labile linkage because there are dehalogenases in tissue. Also, when antibodies or proteins would get internalized, they get degraded to and the metabolite is iodotyrosine, which can exit the cell. So as you can see here, if you have a non-internalizing target and you use standard iodination, it's stays on the surface, your label stays associated with your cells and tissue of interest, you have stable signal in your target tissue, and what's good is that in the organs of clearance and metabolism, your signal goes away. You get very nice images. However, if your target internalizes and you try to radiolabel with an iodine, what will happen is that following internalization, there's metabolism, and the radioactive metabolites will exit and clear. So you'll lose your signal in your target tissue. You still have no signal in your organs of clearance, but it doesn't help if you don't have signal in your target tissue. So in those cases, what you need to do is use a stable or residualizing labeling approach, as illustrated here, where once the antibody is bound and internalized, it will still, the antibody or its metabolites will stay associated with your target of interest. Then you'll have signal in your target tissue, but the price you pay is you will also see either liver or kidney or wherever the radio tracer clears, because the metabolites will be trapped in those tissues as well. So those are things that you have to balance along with everything else that I've mentioned. Okay, so how are we going to do this? How are we going to take mouse antibodies and put them into patients? Fortunately, we do have protein engineering. The biopharmaceutical field is very mature now, so it's very much a straightforward practice to engineer to reduce immunogenicity. One thing we want to do in an imaging agent is to not have it be biologically active. We want inert tracers whose only role is to seek out and engage the target of interest. We don't want to have side effects. We also want to, as I alluded to earlier, accelerate the clearance and the pharmacokinetics, and we'd like to be able to steer where it goes. If it gets out of the blood, we'd like to be able to steer that clearance to organs of interest so that we can keep fields of view clear if we have the choice. And the last thing I'll show is an example of engineering for site-specific conjugation so we can have a more uniform product. So this is what my lab has been working on really for the past umpteen years. I won't put a number on it, but what we've done has been to start with intact antibodies and then just use the variable regions which have the specificity and first of all, by creating single-chain FV, single-chain variable fragments as a building block, we then started building intermediate size engineered antibody fragments so that we have bivalent molecules that have much lower molecular weight than the intact antibody. And as you can see here, typical intact antibody has a slow clearance rate with a lot still left in the blood several days later. And if you move to the fragments, you can see that they have much more, in red, the much more accelerated clearance from the blood, yet they still are able to reach significant uptake in your target tissue by a reasonable amount of time after injection, so that in particular the Minibody and Diabody formats shown here give you contrast, allow you to distinguish between your target tissue and your background within a few hours or a day after injection, instead of having to wait several days for that background activity to clear. You'll also notice that the monovalent fragments typically do not do well because you don't have the benefit of the avidity effect of having two binding sites. What we are trying to do overall is to balance exposure in the circulation and clearance so that you get sufficient exposure to get high enough uptake to get a signal, but rapid enough clearance that you can distinguish signal from background within a reasonable amount of time. And so what we've learned is, as others know, that there's a threshold for rapid renal clearance. So fragments like the Diabody and single-chain Fe will clear quickly via the kidneys. Anything above that size will clear more slowly through the liver. And then in particular, antibodies, intact antibodies, have an added mechanism for very prolonged serum persistence through the FC region of the protein as shown here. And these are all things that we can engineer into or out of our fragments to control their pharmacokinetics. Okay, so I'd like to walk quickly through an example how we've done that, starting with an intact antibody against a target of interest through humanization and then making a fragment and then taking it into further development. So this actually, the example I'm giving you is prostate stem cell antigen, which is a target that's elevated in prostate cancer with low tissue expression in normal prostate, a little bit in stomach. But when prostate cancer arises both in the primary and the metastatic lesions, it's highly upregulated. So that meets the criteria for a possible target. And this top series of images in a mouse with negative and positive tumors, subcutaneous xenografts on each shoulder, show you that if we label the intact mouse antibody with iodine-124 and do serial images, you get very clear distinction of the antigen positive versus the antigen negative tumor. And then we went on to humanize the antibody. We lost some affinity, but we still had respectable affinity. And what you can see is that in vivo, we still had excellent localization to the positive xenograph compared to the negative. But the main point of this slide is that these final time points, imaging time points, MicroPET time points, are at 168 hours, which is a week later. And even at 3 days after injection, there's still all this significant blood pool activity. So this is one of the sort of typical behavior for an intact antibody and what we wanted to address. So instead, we took the humanized antibody, reformatted into engineered antibody fragments. And to summarize with these I-124 images, here's the intact antibody, positive tumor. Here is the Minibody. And here is the Diabody. You can see excellent localization to a target positive xenograft. This is just urine in the bladder. But what you can also see is instead of having to wait 1 week, here is an image done the next day. Here is an image that you can acquire the same day because of that rapid uptake and the rapid clearance, rapid development of contrast. So just wanted to give you then a couple of examples how we're looking towards moving these forward. This is work by a MD PhD student in my lab who just finished. So he had access to this protein. And in addition to, so this is the anti-PSCA humanized affinity matured Minibody. And so he's been doing a series of studies in animal models to understand how can we use this in addition to just detecting tumors. So he's been imaging in knock-in models, transgenic models that have human PSCA, so we have normal tissue background. He's been imaging response to therapy with anti-androgens because this is a target that requires androgen receptor activity for expression, and if you treat with an anti-androgen, that signal should go down. So he's been looking at that. And I did want to show you this one example where he's been imaging bone lesions. So let's back up for a minute and consider how bone metastases are currently detected in cancer. And that's primarily using technetium-99 MDP. Here is what you would typically see in planar bone scans in a patient with metastatic prostate cancer. You can also do SPECT imaging, and with the addition of the three-dimensional and added sensitivity, you can start to pick up even more detail and better localization. And even more recently, many groups have also explored the use of sodium fluoride, F18 fluoride, for bone scans. And as you can see here, that it further increases the sensitivity and the resolution of the lesions that you can pick up in a patient. But the things that you have to bear in mind are that the bone scan is not measuring tumor itself, it's looking at bone remodeling. as a secondary effect of having disease in the bones. So they won't help you to detect primary prostate cancer or metastases in soft tissues. Furthermore, it's non-specific. So the other ways that you can get activity with techniques with bone scans in general could be fractures or arthritis. And it does also complicate the issue if you're using this to follow response to therapy, because frequently there's a flare response. Even if your therapy is successful, tumor cells are dying, there's an inflammatory response, and you get a flare early on, and you still have to wait for that flare response to resolve before you can see what the residual remodeling looks like. So there are challenges with that, and our goal was to be able to directly image the tumors. So this is what Scott did in the mouse model with help from Karen Zhang, another grad student in Lily Wu's lab, with the intratibial injections. These are prostate cancer, 22RV1 human prostate cancer cells that overexpress PSCA and are also marked with firefly luciferase. You can see at 2 weeks after injection into one of the tibia, there's the bioluminescent signal. If you look at the fluoride bone scan, this is what a slice of a fluoride bone scan looks like in a young mouse. You see a lot of activity in the skeleton, in the knees, in the elbows, etc. What you're looking for is asymmetry just below the knees here, and you really can't see much going on at 2 weeks. So at 6 weeks, you can see a very significant bioluminescent signal from the tumor. And here's the bone scan, and those of you up front can see that there's a little bit of a tail below this knee and not below that knee. So that asymmetry is the positive bone scan in this young mouse. Now, if we switch to the antibody-based scan, I think you may need to be in the front to see this, but even at 2 weeks, if you look at the left tibia here, maybe it's the right tibia, left is right, right is left. The right tibia here, there is signal inside the bone that you can see on the coronal and sagittal slices. And then by 6 weeks, it's really clear. You can see that really, really strong signal coming from that knee. So this is the difference between a more general approach towards looking at bone remodeling to specifically targeting the tumor with an antibody-based agent. Okay, so where that stands now is that through a lot of effort, I've already went through briefly through the steps of humanization, affinity maturation, in collaboration with researchers at the City of Hope, including David Coulter, who's here. We went through the process of CGMP production of this fragment, filed an IND, got that approved. The goal was to conduct a clinical imaging study comparing I-124 PSCA minibody imaging to conventional imaging, bone scan and CT, in men with metastatic prostate cancer. And that study actually initiated last week. So I don't have any images to show you, but we have a couple of clinical studies going on now, and that's been the culmination of a lot of work, but very exciting to be at that point. Just quickly wanted to talk a little bit more about Diabodies for same-day imaging, because for a number of reasons, we'd like to be able to do imaging within an hour or two of administration. Certainly for clinical workflow, that would be much better than having to come back a week later or a day later. Furthermore, we'd really like to be able to explore the possibility of radiolabeling these antibody-based fragments with fluorine-18. That's the most widely available PET isotope, and it really has excellent physical properties and half-life. So the Diabody actually, though, if you look at the pharmacokinetics, it reaches peak uptake at 1 to 2 hours after injection with a 3 or 4 hour half-life. So those kinetics actually should match very well with fluorine-18. And through the years, with a variety of collaborators, we've been pursuing labeling Diabodies with fluorine-18. On the chemistry side, we've been moving towards streamlined one-pot syntheses. We've moved towards microfluidic platforms to be able to do this on a more routine basis, because what we really want to be able to do is to radiolabel these Diabodies with fluorine-18 on a routine basis. Right now, it's more still bench scale. But what I can show you is that we've been able to show, at least in 3 different antigen systems, that we're able to take Diabodies with specificity for CEA, HER2, or PSCA, radiolabel with fluorine-18, and acquire images in mice within 1 to 4 hours after injection. So I think that really shows the feasibility of this approach. Okay, and one other addition that we've done has been to introduce cysteine residues for site-specific conjugation. This becomes important. The smaller and smaller your fragment is, you really want to move away from doing random radiolabeling, because you might damage the protein and interfere with its immunoreactivity. So here is actually an anti-alcam diabody where we've added cysteine residues to the C-termini, and they actually don't just hang out there, they form a disulfide bridge. You can label this randomly on lysine residues with the chelating agent DOTA, and when you examine the product, you will find, because it's random, you find a spectrum distribution of species with 4, 5, 6, 7, or 8 DOTA moieties attached. Or you can take that same protein, lightly reduce it with TCEP to release these reactive thiol groups, use a maleimide approach to attach your chelating agent, and then when you look at this by mass spec, you can see each of these polypeptide chains has exactly one DOTA moiety, and it's on the C-terminal cysteine. So this really is an approach that we can facilitate through protein engineering that enables site-specific oriented conjugation with preservation of immunoreactivity, and it gives you a very uniform product, potentially. Okay. And I also wanted to mention then this opens the door to all kinds of cargo that you can add to your antibody. I've showed you DOTA maleimide for radiometal labeling. We've done small molecule fluors, such as Alexafluor dyes. We can use this to conjugate to nanoparticles. We've actually done quantum dots, cross-linked polyliposomes. We've collaborated with a variety of laboratories to add these as targeting agents to their nanoparticles. And by having the Cys-Diabody, we can we have a very small agent. It's only 5 by 7 nanometers as opposed to an intact antibody, and we can put it on the particle in an oriented fashion so that their binding sites are facing outwards. So again, a smaller and more uniform approach. And of course, we've been working on labeling these Diabody site specifically with fluorine. I did want to show you one quick unpublished example of one thing you can do, coming back to the fluorescent dyes. So in collaboration again with Rob Ryder and Jeff Sohn, a clinical fellow in his laboratory, we put Cy5 onto an anti-PSCA specific Cys-Diabody and used it to image fluorescently and intraoperatively tumors embedded in a mouse model. So that's an area that many groups are pursuing the potential of using fluorescently labeled antibodies for intraoperative use, and I think that this format actually lends itself very well to that application. So it is a platform. You can see that over the years in my lab, we've been taking antibodies with a variety of starting specificities. There's not a complete list. And you can reformat into minibodies or diabodies and reproducibly get yourself a PET agent that is specific based on cell surface phenotype of your target. So typically 18 to 20 hours to get a minibody image, 1 to 4 hours for a diabody. And just briefly, there are many ways we can use this thing. I'm not going to give you, I have examples, but I'm not going to go through them in detail here. But the kinds of questions you can ask once you have a general approach to imaging based on cell surface phenotype include, in addition, just simply is there a tumor there or not, you can learn about the phenotype of that cell population, including tissue of origin. Is it lymphoid? Is it from prostate? Where is it from? You can look at things like activation markers. You can look at elevated expression of receptors. For example, HER2 overexpression is a question that many labs are addressing. You can use that information to drive, looking at target expression to drive therapy selection. You could directly assess the delivery of your therapeutic by labeling your therapeutic. You can look at target occupancy by essentially doing competition studies in vivo. You can evaluate internalization and catabolism in vivo, perhaps by comparing the uptake, metabolism, and clearance of your biological labeled with stable versus a residualizing versus a non-residualizing label. You start to query mechanism, direct or indirect, many ways you can look at response to therapy and quantitation. And so those of you who are interested in exactly what kinds of things you might want to image and how, there's a session later today on cancer biology where, for example, you go through how you can image the 6 hallmarks of cancer, and then you can think about how you can actually use antibody-based probes to do exactly that. And then the last example I wanted to go through, sort of pushing the limits, is that we can also start to look at immune cell subsets. I think this is a really, really important area for future imaging because immune responses are involved in so many diseases. In addition to oncology and all of the very promising immunotherapy approaches that are now emerging in oncology, immune responses are key to cardiovascular disease, Alzheimer's, diabetes, autoimmunity, et cetera. And we have right now FDG, which is very good for imaging immune responses, but it's nonspecific. What we'd like to be able to do is find a way to specifically image components of the immune response by looking at immune cell subsets. And again, the antibody approach lends itself to exactly that. So I'll just show you one example from Richard Tavares. He presented this last year at WMIC, where he's looking at CD8 in T cells. He's worked on a couple of antibodies, one which is specific for one allele that's in some strains of mice, and another one that is pan-specific for all CD8 alpha in mice. He's developed Minibodies labeled with copper-64, and in this initial experiment showed that this 2.43 Minibody, which is specific for the allele that's present in black 6 mice, shows you the spleen and the lymph nodes in the black 6 mouse, but does not pick them up in the C3H mouse, which I think is a great demonstration of the exquisite specificity of antibodies, because the only difference between these two alleles is a single amino acid variation. And he subsequently went on and then did some further studies in the normal black 6 mouse, where he either blocked the CD8 on the T cells in the normal mouse by pre-administering cold Minibody, or he did a depletion study where you can take an anti-CD8 antibody, treat the mouse for 3 days ahead of time, deplete all its CD8 positive T cells, and then he imaged and showed that indeed he had manipulated the mouse, the spleen is gone, the lymph nodes disappear. So we're imaging endogenous T cells in a normal mouse and can now start to look at immune responses in a normal mouse. So this is just the quantitation. So I'll wrap up here, because I think I'm out of time, and just remind you that what I hope I've shown you is that antibodies provide a terrific source for imaging agents, and in particular, there are so many existing, optimized, well-characterized antibodies out there, developed for therapeutic purposes or whatever, and to convert those into a engineered antibody for imaging purposes is, for the most part, very straightforward process. You can also generate imaging agents from new novel antibodies as well, such as the antibodies that we get from human phage display libraries or transgenic mice. So basically, you have huge spectrum of antibodies and specificities available, and that we can make these Minibodies or Diabody formats, and they have all these properties that are highly suited. We can really tailor them for imaging purposes. They're non-immunogenic, biologically inert. They demonstrate rapid targeting and imaging. We can either direct the clearance to the liver or kidneys, depending on where we want to image. Because they're smaller, they do have improved tissue penetration over large intact antibodies. We can engineer them for site-specific conjugation, and particularly with the Diabody format, we can look at doing same-day imaging with fluorine-18 labeled proteins. And as I mentioned, there are diverse applications possible in oncology, immunology, and many other diseases. So I'll wrap up there. I'd like to thank all the current and past members in the lab. I've highlighted in blue many of the people that contributed directly to the work that I've shown here. And I'd also like to point out that we have 3 oral presentations if you want to hear more about imaging B cells, human B cells, mouse T cells, or CMET in oncology applications. And this is Koyu here, this is Kirsten here, and this is Richard here. And they have talks later on in this meeting, and posters as well, which we'd love to have you visit. Thanks.